take it over, Ella?
Sure. Just quickly, I am Ella Shuleta. I'll be moderator for this session. And all speakers, when you will get to 10 minutes, I will tell you about that. So we are already at 1 p.m. So Travis, go ahead and start whenever you're ready.
Great, thank you very much, Ella. Can everyone hear me okay? Yeah. All right, so this is a literature synthesis meta-analysis study that I've been working on since about the time we all went into quarantine last March. And the focus of this study was really to look at how management practices and environmental factors impact the ability of corn-derived crop residues to be transformed into soil organic matter and soil organic carbon and be retained in those soil environments. So before we delve too deeply into that, first let's start out talking about how soil is the largest reservoir of carbon. So given our global climate crisis that we're facing, there's a lot of interest in this question about how we can manage soils such that they are actively acting as sinks for atmospheric carbon dioxide. That is, they're pulling carbon dioxide out of the air and then storing it below ground. And in trying to answer this question, there's been a lot of research evaluating how the environment and how implemented strategies affect the turnover and accumulation of soil organic carbon in soil. Now, this list here is by no means comprehensive as far as environmental and management factors that could impact turnover, but we have seen throughout these factors wide variations in terms of positive or negative impacts on the soil organic matter present in soil. So the goal of this study was really to use data present in published literature to help us develop a global perspective and answer this question about how soil organic carbon stocks change when land use is converted into a continuous corn monoculture. And we also wanted to address how management strategies impact those changes that we see in those systems. So before I get into my hypotheses, I just have a couple of housekeeping things that I would like to cover. You will probably hear me interchangeably use the term soil organic matter and soil organic carbon. While soil organic matter comprises all elements, such as carbon, nitrogen, phosphorus, we know that soil organic matter is really dominated by carbon. So the data you're going to see presented is all in terms of carbon, but you still might hear me change the terms used as far as soil organic matter and soil organic carbon. When you hear me talking about inputs, outputs, and retention, the inputs are not a controlled rate of carbon input. This is instead rather the quantity of corn-derived carbon that it has the opportunity to decompose and become soil organic matter just from crop residues being left in field. And outputs would be losses of mineralization of soil organic carbon inputs. And the retention would be the inputs of corn-derived carbon retained as soil organic matter divided by the total quantity of inputs that are entering the soil environment. So lastly here, you'll also hear me talk about later on soil organic carbon pools. We'll talk about fast and slowly cycled pools. These fast cycled pools are pools of soil organic matter that are actively used and cycled by microorganisms, whereas these slowly cycled pools are more persistent forms of soil organic carbon. They're not as readily used by microorganisms for their own nutrient acquisition and biological processes. So the hypotheses that we set for this study was firstly that when corn-derived carbon inputs to soil are low, the retention efficiency should be relatively high. As far as soil goes, we hypothesize that fine textured soils and unweathered soils should be able to retain corn-derived carbon more efficiently than coarser textured and more weathered soils. And in terms of management, we hypothesize that nitrogen fertilizer additions and residue removal would increase the retention efficiency of corn-derived carbon as soil organic matter. And we hypothesize that tillage had that opposite effect. So as I mentioned, this was a literature synthesis type study. So the majority of the methods were really acquiring data that we deemed appropriate. So to do that, we utilized the Web of Science database and the Google Scholar search engine with these two key phrases. And whenever search results came about, we deemed them appropriate to include within our data set if they reported these three specific measurements: total soil organic carbon, corn-derived soil organic carbon as estimated specifically by the natural carbon-13 abundance method, and the quantity of corn residue carbon inputs throughout the experimental duration. Now, I do want to mention that the majority of the work for this study was really standardizing data and making sure that we present our data in all equal terms. As you can imagine, literature that spans a couple of decades reports things in different units. So that's where a lot of the work came in with this study. Unfortunately, if I was going to cover that, we would be here quite a while. So if you do have questions, please feel free to ask me at the end. But for now, I'm going to jump right into the results and discussion. So what we found throughout our literature search was that we had 24 viable studies that yielded 111 data points that we deemed appropriate for our analysis. And the red circles on this map indicate where experimental stations were located within the publications we deemed appropriate. And so the data from these studies first showed that when you convert an ecosystem into a continuous corn crop, there is an initial 9.5% decrease in the initial soil organic carbon stock. And for every year that that system remains in a continuous corn system, you continue to decrease that stock by 24% annually. Now, to be completely transparent about this data, this regression model is not statistically significant, but I think it's really important to keep in mind, especially given the nature of this data, that p-values are not everything when it comes to meaningful data. So when we superimpose a line at 0, which would indicate that there is a stable soil organic carbon pool, we see that the majority of these data points are falling below that 0 line. So even though this model doesn't achieve statistical significance, I think this is evidence to suggest that if you're going to convert into a continuous corn cropping system, there could be some negative implications for the initial state of the soil organic carbon pool. Now, to more directly address our first hypothesis that high input rates should yield lower retention efficiency, we see here that there is a relatively steeper slope between 0 and 100 megagrams of corn residue carbon added compared to once you pass that 100 megagrams per hectare of carbon. So this is somewhat reaching a plateau at some point in time. And what you see here between the 0 and the 100 is probably indication that you have corn-derived carbon entering a quickly cycling soil organic carbon pool. So basically you have native carbon that is actively being cycled by your microorganisms. As they cycle that out, they're opening up exchange sites that can then retain corn-derived carbon. But once you move past that, we start seeing carbon cycling in these more slowly cycling pools. So the added corn carbon does not necessarily become incorporated into those more slowly cycled pools. And that's likely why we see this lower retention efficiency at a high input rate. So this is further exemplified when we look at annual input rates for each study. So we see here that the proportion of corn carbon inputs retained on an annual basis is much greater when inputs are low compared to when those inputs are much higher. And in terms of soil properties, we see that coarse textured soils retain a significantly lower quantity of corn-derived carbon compared to these more medium type textured and finely textured soils. So your medium textured soils would be your more loamy types, and your fine textured soils would be your more clayey soils. So intuitively, you might think that clayier soils should retain more soil organic carbon than a medium textured soil, but it's important to keep in mind the weathering status of that soil. So what we see is that as weathering increases, you have a decreased retention efficiency. So it's really important to keep in mind that not all clays are equal. Just because you have more clay doesn't necessarily mean it's a more reactive clay that has the ability to retain more soil organic carbon.
You're close to 10 minutes.
Thank you, Ella. So in terms of management practices, this is where we employed some meta-analysis techniques. So basically what we see is that in terms of nitrogen fertilizer, tillage, and the retention or removal of crop residue in field, we see no significant effects of management on the ability of corn-derived carbon to actually become incorporated and accumulate as soil organic matter and soil organic carbon. Now, I think it's really important to keep in mind here that this is a global perspective. As I mentioned, there have been a wide variety of positive and negative results reported throughout literature. So given a more diverse and broad data set across the world, we might be able to more effectively pin down what is happening at the local and regional scale. So that's something to keep in mind moving forward. And all in all, we found evidence to support our first and second hypotheses, and we rejected this third hypothesis because we found no evidence to suggest that management factors are actually significantly impacting the retention of corn-derived carbon as soil organic matter. So moving forward, we would like to improve the robustness of that meta-analysis by bootstrapping those confidence intervals. And we intend to expand on this data set by adding some novel data that we have collected from continuous corn plots at Spindletop Farm. And with that, I'd like to say thank you to everyone for your attention, and I'd be happy to take any questions. I'd love to hear any feedback that you all might have.
Thank you, Travis. So anyone, if you have question, feel free to unmute yourself or write question in a chat box. There are already 2 questions. I'm gonna go ahead and read them for you. When you say carbon added, does that imply residue incorporation or carbon dropped at the soil surface?
So I think that either one could be appropriate, since we looked at studies that both removed residue and allowed residue to sit on the field. So it's really a measure of what the maximum possible amount of carbon that could have entered the soil. So many of these studies, actually all of these studies, have accounted for root biomass below the soil surface as well.
Okay, there is another question. How do you suggest that producer, no, I'm sorry, there was a previous, another one. How did you define weathered versus unweathered? Also, were these COC measurements all taken from the same depths or standardized for depth?
Yeah, so as far as the depth standardization, as you can imagine, the literature really reports a wide range of soil collection depths. So we standardized everything to only account for data that is reported within the upper 10 to 35 centimeters. So that's really what we're focusing on here, is accumulation within that range of soil depth. And I'm sorry, but could you repeat the first part of that question again, Ella?
Yeah, it was, how did you define weathered versus unweathered?
Right, so weathering, if there is a figure actually in the Elements and Properties of Soil by Weil and Brady that has a weathering, I guess, a degree of weathering based on soil order. So we actually were able to characterize the soils by their soil order from the publications and then classify them as weathered or unweathered based on the diagram from that textbook.
There are 2 more questions. How do you suggest that producers test their soils to assess the degree of weathering / potential to store carbon through management practices?
Yeah, that's a really good question. The one thing that I could say, this is really probably not feasible, I think, at a producer scale. I think understanding the mineralogy is going to be very telling about the potential for future capacity to store more carbon. So I think that will probably be the most reliable measure of potential, but something that's simpler that could be done more at the farm scale would probably simply measuring the existing soil organic carbon stock to see how inundated the soil is with soil organic carbon prior to any type of management or cropping systems.
Mhm. There is one more question, Travis, but for the sake of time, it would be great if you could answer it in a chat box.
Of course, thank you.
And thank you so much. And we will be moving to the next speaker, who is Anik Mahmoud. Go ahead and share your presentation. Okay, and we can see your PowerPoint, so go ahead and start whenever you're ready.
Are you all, good afternoon. Are you all hear me okay?
Yes, yes, we can hear you.
Okay. First of all, I want to say good afternoon to all. And today I'm thrilled to share my research funding of my MS project entitled Selenium Removal and Pollution Swapping Potential of Passive Anaerobic Reactors Receiving Cold Slurry Impoundment Awards. So my project revolves around the remediating wastewater contained in cold slurry impoundments, which are very common in Kentucky and West Virginia and other Appalachian states in the USA. Here you can see a picture of impoundments that is surrounded by dams and filled with cold slurry waste, which is actually the byproduct of coal processing process that removes silt, clay, and trace elements. For example, in coal, we typically found minerals like ferrous selenide, and this is, as you can see, but it can hydrolyze into selenate. These liquid wastes are usually stored in multi-billion gallons impoundments in impoundments, and here you can in USA there is around more than 700 impoundments present. So why do we care about the giant structure? Here you can see sometimes these impoundments are built very near to the environment where we live, and this is very threatening because this cold slurry contains elements, for example, selenium, iron, arsenic, in higher concentration than EPA recommended. For example, selenium, like for example, EPA recommended that selenium should have less than 3.1 ppb for aquatic, warm water aquatic habitat, for lotic system especially. And this hazardous waste sometimes chronically leaks through the dams to the adjacent streams, posing threatening to the sensitive species like fish. Here is a photograph of selenium-induced toxicity in fish. Here you can see that sunfish and creek chub has malformation, like sunfish has facial malformation and creek chub has spinal malformation. So how do we reduce this selenium-induced toxicity? A possible biological approach to reduce selenium concentration in cold slurry is passive anaerobic bioreactors. Here you can see that 4 steps of making passive anaerobic reactors in field condition. Generally, it is a line trains filled with organic substrates, and where wastewater has been pumped by using wires and gravity. This technique, this treatment process has been used for remediating nitrate for more than 20 years by the process of, as you can see, this red line, that by the process of denitrification. It has some advantage. For example, this treatment process do not need energy, has minimal wildlife exposure, and also thermally insulated. The similar approach can be taken to remediate selenium by passive anaerobic bioreactors and by promoting the dissimilatory selenium reduction. So to illustrate more the dissimilatory selenate reduction, here you can see the red dots. It's a two-step process where selenate is reduced by the enzyme selenate reductase in the periplasm of the microorganism to selenite, and then selenite is reduced into elemental selenium by selenide reductase, and then get out of the bacterial cells. Here is a photograph. In the middle of the photograph, you can see that the formation of red color elemental selenium, which is sparingly soluble and non-bioactive solid. However, the story doesn't end here. The PAB system has some disadvantages, which is pollution swapping, which is normally not considered during making these PAB reactors. Actually, to further explain more about the pollution swapping, which is the production of dissolved nutrients, organic carbon, trace elements, and greenhouse gas from the organic substrates in PAB system that potentially can have detrimental effect to water and air quality. I hope that the background information I gave brings some appreciation to my project goals, which is I have two project goals. One of them is to evaluate the capacity of PAB system with different organic substrate to remove selenium from coal slurry, and also to evaluate the pollution swapping of PAB with different organic substrates. And my hypothesis is that these two goals is significantly affected by the types of organic substrate in PAB systems. To achieve this goal, I did a microcosm study where I built passive anaerobic reactors by messenger filled with different organic substrate and simulated coal slurry. That 4 types of organic substrate has been used, which is wood chips, Swissgrass, and miscanthus, and Timothy hay. And water, I monitored the chemical changes in water for 7 months. And to further illustrate, to further explain more from solution, I monitor pH, EH, and also selenium and other trace elements by ICP, OES, and MS. Thank you for Dr. Andrian for helping me on that. And also DOC, ammonium, nitrate, sulfate, and phosphate. And I also monitored greenhouse gas by using gas colorimetry. And I did the statistical analysis determined by ANOVA and 2K. And I also wanted to monitor the effluent, whether it is good or not to release into the environment. I compared my measured concentration parameters with the pristine system concentration, and I calculated the water quality index. To further explain more the calculation of water quality, the water quality index, I want to say here you can see that I have selected some pristine parameters that I want to compare with my measured parameters. And to explain more, here you can see the calculation that I used to calculate the water quality index. Is a function of three. It's a function of 3 parameters. So you can see the 3 variables. F1 is related to the number of parameters that fail, and F2 is related to the number of total failure during the experiment, and F3 related to the extent that parameters deviate from the pristine concentration. So this water quality index has a very good advantage because it allows me to express the different measurement I did in single metric. Here you can see the water quality index score interpretation. If the score is 0 to 4, I will say this is the poor quality. And if the score is 95 to 100, and I will say the effluent quality is excellent. Here I want to share some of my findings. Most of the chemical constituents that I measured were significantly affected by the types of organic substrates in the PAB. For example, here you can see the EH. The EH is declined rapidly in all PAB system, except the wood chips. But however, in day 15, the EH raised to minus 200 MV in all the reactors. Here is the example of DOC release from the substrates. Here you can see that the Timothy hay has the significantly higher release of a DOC in the system. So here the selenium reduction, I am showing here the selenium reduction rate over time. So you can see that in month 011, the selenium reduction rates were significantly faster in PAB with Timothy hay, and followed by miscanthus, which is equally good as miscanthus, the Swiss grass, and the wood chips has the lowest reduction rate. During a 1 and 2 month and the 3 month, the selenium reduction rates is generally decreased. But however, in the 3 or 4 month, the wood chips performs better than other substrates. In day 30, you can see that 95% of the selenium has been removed. But however, that the concentration I get is about 30 times higher than EPA guideline. Not only the other chemicals that I have just discussed, other chemicals are also have significantly affected by the substrate that I used. Like for example, here you can see the wood chips has lowest pH, and Timothy hay has the highest EC, DOC, and phosphate, and also the carbon dioxide emission. The water quality indexes that I calculated, I see that most of them, all of them are in between the range 7 to 9, which is very poor quality. And results put into question whether this PAB system, including the denitrification reactors, are good enough for long-term, for short-term application. So longer-term experiments are required to determine if this WQI would improve with time. So this is my one of the recommendation to monitor more to see whether this WQI value is getting up or not. The conclusion is that I would draw that selenium removal rates and pollution swapping in PAB were significantly affected by the types of organic substrate and maturation time. And this study also emphasized that evaluating both primary and secondary pollution swapping of the PAB before considering the implementation in the field scale. Thank you so much. If you have questions, I'm happy to answer. Thank you.
Thank you. We already have one question in a chat box. Why do you think hay releases so much more DOC and CO2 than other substrates? Is there a reason why it's degraded so quickly?
Okay, I did the proximate analysis of all the substrate before implying into the PAB system, and I see that the hay has larger water-soluble carbon, and it has less lignin content. That's why the DOC limit is so high in PAB system, especially the Timothy hay and Swiss grass. Yeah.
Okay, we still have some time for more questions. Actually, I was wondering when you were describing your bioreactor, and you said that you used slurry substitute. Why you were using a substitute, and what is the substitute actually?
I mean, you are saying the synthetic coal slurry. Why I'm using synthetic coal slurry? Oh, I try to get the coal slurry, but the company and the coal processing plant did not allow me to get this slurry because they don't want that. So I use my publications, and I make the synthetic coal slurry, which is consistent with what we found. We will find in the nature system. Yeah.
I thought so. It might be due to logistics reasons.
Because they don't want to maybe to show that they are doing harm by using this coal, by promoting coal and degrading the nature environment. So they don't want to share their information about that. Maybe that's why.
That makes sense.
Mhm.
All right, thank you.
Welcome. Thank you for all for your patience. Good luck all.
Thank you. Our next speaker will be Jared Cochran. Go ahead and share your presentation.
Says I need to,
Oh wait, because yeah, Anik was still sharing his.
yes.
All righty.
All right, everybody see it?
Yep. Go ahead and start whenever you're ready.
Okay. Hey, everybody. My name is Jared Cochran. I'm a PhD student for Olga Tusko. And today I'm going to be presenting on some of my research on the application of iron oxide nanoparticles against infections induced by drug-resistant pathogens. During this presentation, I'll be giving a brief background on antimicrobial resistance, also known as drug resistance, and why nanoparticles can be an effective tool against antimicrobial resistant spread. I'll also be going over a few of the methods I'll be employing during my research and some of the results I found so far. Okay. As many of you know, antimicrobial resistance is a growing public health concern. The World Health Organization has predicted that by the year 2050, over 300,000 people in North America alone will die due to complications related to antimicrobial resistance. The spread of antimicrobial resistance is due in a large part from the overuse of antibiotics, not only in medical settings, but also in the agricultural industry. Antimicrobial resistance threatens not only human health, but also our modern agricultural system, which employs antibiotics in a lot of their practices, especially in cattle rearing. And indeed, we are already starting to see the effect of antimicrobial resistance in the dairy industry. Bovine mastitis is one of the largest expenses in the dairy industry, costing billions of dollars each year. Bovine mastitis is an infection of the mammary glands, and during the infection, the cow is unable to produce milk. Therefore, the farmer loses all product during that time. The infection is quite difficult to treat, with failure rates up to the 80% in some cases. Although a lot of pathogens cause bovine mastitis, Klebsiella pneumoniae and Streptococcus aureus are notable pathogens because of their ability to rapidly gain antimicrobial resistance. To combat these two pathogens, new strategies to treat the infection are needed. Recently, nanoparticles have gained a lot of interest as a new source of antimicrobials. This is due to a lot of nanoparticles having antimicrobial properties naturally, and these properties can be enhanced through altering the surface coatings. Additionally, magnetic nanoparticles are able to kill pathogens within an alternating magnetic field. The magnetic field can cause the particles to vibrate, and either through an increase in temperature or through physical active vibration can tear the bacteria apart. For my project, I'll be using an iron oxide nanoparticle. We chose this particle because it is ferromagnetic, meaning that it can be affected by a magnetic field, so that we can employ the alternating magnetic field strategy. It's also considered a greener antimicrobial. This is because its composition, magnetite, is naturally found in soils. And finally, iron oxide nanoparticles are naturally antimicrobial through the Fitton reaction. This is a process of converting hydrogen peroxide to hydroxyl radicals. Hydrogen peroxide is naturally produced during metabolism, so it's typically found in the environment where bacteria grow. Additionally, although iron oxide nanoparticles typically have a negative charge in the pH range that we're interested in, the coating can be, the surface can be coated with Chitosan, which is the naturally occurring biomolecule, which will give the particle a positive charge. This will better let it adhere to the cell surface, which is also typically negatively charged. And once adhered to the cell surface, the Fitton reaction could be able to produce hydroxyl radicals that can break down the cell wall of bacteria. Okay. Am I still there?
You are, but you're not sharing your screen anymore. You have to start sharing again.
I don't know why that happened.
Oh, it's here. It's there.
All right, oh, that's weird. Okay, for my research, I'll also be using C. elegans. This is a common model species for toxicity and antimicrobial screenings. This is because the nematode is able to grow from an egg to an adult within 3 days. Additionally, C. elegans shares a 90% genetic similarity with mammals in the case of their innate system. There's also numerous genetic tools used to measure the genetic effects of exposure in nematodes. And finally, and importantly for this study, C. elegans is susceptible to Klebsiella pneumoniae and also Staphylococcus aureus. So they can actually be infected with the pathogen and will die from the infection. So the objectives of this study are to assess the toxicity of iron oxide nanoparticles to C. elegans, to determine the antimicrobial activity of iron oxide nanoparticles and its ability to treat infections, to determine if alternating magnetic field improves infection recovery, and finally investigate the effects of nanoparticles on the molecular mechanisms of infection and recovery. The particles that we'll be using were obtained from a collaborator in Michigan State University. Briefly, the particles were produced through dissolving iron 3 chloride with sodium acetate and chitosan and ethylene glycol. The solution was then heated at over 200 degrees Celsius for 8 hours and then allowed to cool for overnight. The particles are separated from ethylene glycol with dialysis, and then the particles are suspended in water. Right now, the size isn't really known, but we'll be finding that out through transmission electron microscopy. For a toxicity assessment, I will be exposing C. elegans to increasing concentrations of iron oxide nanoparticles until either mortality is observed or there is a decrease in reproduction. The concentration below that threshold will be used in the antimicrobial assessment. That way I'll know if any of the negative effects I see in the nematodes is from the infection and not from the nanoparticles. For the antimicrobial assessment, it's pretty simple. I'll just be taking nematodes and put them in their own individual well and exposing them to either Klebsiella, Staphylococcus, and or an iron oxide nanoparticle. If the treatment is effective, the nematodes will live and they'll maintain, they'll be able to still move around in solution. But if the treatment is not effective, the nematodes will die, and due to osmotic pressure, they will get this rod-like shape. Okay. So my results so far, I have done a toxicity assessment, and I have found that the nanoparticles do not cause mortality in the nematodes after 24 hours of exposure. The y-axis shows % alive, and the bottom shows the control and the concentration. As you can see, the control, which is in the blue color, and the other exposure concentrations are all very similar. Even though the 100 milligram per mil and the 150 milligram per mil seems less, it's not significantly different. I've also done the reproduction assays where I've exposed nanoparticles, I've exposed C. elegans to the nanoparticles and measured the number of offspring produced. The y-axis shows the number of offspring produced by the nematodes over a 3-day period, and the bottom shows concentrations. As you can see, the control and the concentrations do decrease compared to control, but only at the 150 ppm range do I see a significant decrease in reproduction. So therefore, for my antibiotic exposure experiments, I will not be using 150 milligrams per ml. I've done one study on exposing nematodes to Klebsiella with 50 milligrams per liter and also 100 milligrams per liter. It should be liter, not mil. As you can see, the y-axis is % alive, and the bottom is the number of hours from exposure. The control, we had a very little decrease in live over the 3-day period. But when exposed to Klebsiella, you can see that the percent alive decreased, with the the fewest being alive with just Kleb exposure. Though the Kleb and the iron oxide exposure at the same time had a higher percent alive compared to just Kleb exposed, it's not significantly different.
10 minutes.
Okay. So for the future of my work, I will continue to do Klebsiella and Staphylococcus exposure experiments with iron oxide nanoparticles. I also will be assessing different coatings, specifically cysteine and one that's a bacteria-specific receptor. I'll also be assessing the effects of an alternating magnetic field on the treatment of C. elegans infected with Klebsiella and Staphylococcus. And finally, I'll be investigating the molecular mechanisms of recovery. Okay. For acknowledgments, I'd like to thank Olga Tusko and Jason Unrin. I'd also like to thank Vanjie, Tristy, Tammy, Catherine, and Mark Coyne, and Freytag. Okay, if you have any questions. Thank you.
Thank you, Jared. We have plenty of time for questions. And we already have one. Can you explain the magnetic field thing? What you are expecting there?
So with the magnetic field thing is that we'll have a magnetic field and we'll alternate the magnetic field. And this will cause the particle to vibrate rapidly depending on the rate of the altering field. And this will increase the temperature around the particle, which can kill the bacteria, or if the particles adhered to the bacteria, can actually physically sort of like rip the cell wall apart. It's called hyperthermia, but they're not quite sure if it's actually because of the physical vibration or if it's because of the increase in temperature that actually kills the bacteria. So that's one of the things I'm hoping to be able to find out in my study.
Okay, we still have time for questions. If you want to ask something, you can unmute yourself or write it in the chat box. I was actually wondering when you were speaking about the differences in offspring number on that figure. Was it because you think that was, yes, this one. You think that was because some of the individuals became infertile, or all of them were laying just less eggs?
I'm not quite sure why there was a decrease in reproduction. I suspect what happened was that the particles caused the bacteria in the solution with the nematodes to aggregate, and I think it made it more difficult for the nematode to eat. So there was less like energy available for reproduction as compared to control. Okay.
There is one more question. Why did you choose mastitis to work with?
It's a very difficult infection to treat, like I said, up towards like 80% failure rates in a lot of cases. And we have some ideas about how it might be applied. And we think that the alternating magnetic field might be able to really specifically help with that, because mastitis, the bacteria create biofilm typically in the mammary glands, which is very difficult to penetrate. But with the magnetic field, you can either pull the particles through that biofilm or disrupt it through the alternating magnetic field. And so we just suspect that there might be a lot of potential there.
Okay, we still have a little bit of time, maybe for one question. It looks like we can slowly move to the next presenter. So Jared, if you will stop sharing your screen. And the next presenter is Tim Shaw. Thank you. Tim, go ahead and share your presentation with us. And we can perfectly see.
Okay, can everybody hear me okay?
Yes.
Cool.
Go ahead and start.
Okay, so good afternoon, everyone. Today, I'm going to be talking about a research project that spawned about a year and a half ago. I was using the neurotransmitter dopamine as a control treatment in an experiment, and I noticed that dopamine seemed to elicit a strong and consistent physiological response from plants. So I was expecting it to be relatively biologically inactive, so there was a little bit of a surprise for me. And after a little bit of reading, I found that dopamine and a lot of other biogenic amines are actually widely present in the plant kingdom. Their activity in plants and their presence has been the subject of a number of different research papers since about the late 60s. But the physiological activity of these compounds are not super well understood. So today I'm going to talk about my attempt to shed a little bit of light on the activity of these compounds in higher plants using an Arabidopsis forward genetic screen. And I'm also going to talk a little bit about how I plan to use next generation sequencing in order to identify the mutations that I find in that screen. The first things first, a little bit of background. So the general catecholamine biosynthesis pathway, which is here on the right of the screen, exists in some form or another in a lot of different facets of the plant kingdom. What I found really interesting was that a lot of these compounds have the properties of allelochemicals. So these are compounds that basically plants secrete that reduce the growth of surrounding plants. So L-DOPA in particular, the precursor of dopamine, there's a lot of evidence that that is something that happens. So for such a widespread pathway, the physiological function of a lot of these compounds aren't very well understood. But in the case of dopamine, it seemed like a lot of the roads appeared to point towards hormone signaling. So that kind of put the research a little bit in the purview of our lab, because we do a lot of hormone signaling research in our lab. So this is what plants treated with dopamine look like. So this is 2 weeks worth of growth. This is just Columbia 0 wild type grown on 0 or 1 millimolar dopamine. And the typical response is you get a reduction in rosette size and an increase in primary root growth. So this kind of a physiological response is pretty good fodder for a forward genetic screen to try to identify point mutations that bring about resistance to the compound. So the basic idea of this is that you take a population of Arabidopsis seeds and you treat them with a chemical mutagen such as EMS in our case. And EMS, depending on the strength of the compound and how long you incubate the seeds for, will introduce about 100 to 2,000 point mutations per genome. You then go ahead and screen that population for your phenotype of interest. In my case, it was dopamine resistance. So here on the left, we have the response of the wild type. So whenever you grow it on 0.3 millimolar dopamine, you get this shrunken rosette. And to the right of the wild type are the dopamine resistant mutants that I have isolated so far, or at least it's just a couple of them. Of particular interest so far has been DAR1. Dopamine appears to actually stimulate the production of chlorophyll or the biogenesis of chloroplasts. So that was pretty interesting. But having these mutants aren't particularly useful unless you identify the underlying causal mutations for the phenotypes. So traditionally, identifying the mutations of these mutants would be a very labor-intensive process. It could take several years, but thanks to advancements in whole genome sequencing, it can be done in under 6 months if you're motivated enough. So basically, the protocol looks a little bit like this. You mutagenize your seeds, you isolate your mutant, and then you backcross your mutant with the parental ecotype. In this case, it's WS. You get your F1 generation, and then you self your F1 generation. And in the case of most of my mutants, they are recessive. So I end up in the F2 segregating population having a situation where for every 3 plants that are susceptible to dopamine, I get 1 plant which is resistant to dopamine. So this is really step 1 of mapping the underlying mutations. And this is where I'm at right now with the project. I've got F2 segregating populations for a handful of different mutants, and I hope to proceed to these next steps here in the coming months. So the first thing that I plan to do is I first have to get a good reference genome. Thankfully, working with Arabidopsis, it's a model plant. It's really, really easy to get. But just for a little bit of background, how these things are generated, you take the genomic DNA, you shear it into, depending on the sequencing platform, smaller pieces. Very often it's 100 to 150 base pairs. And then you sequence these short, these little short pieces of DNA, and you get back this big data set, which you reassemble computationally. Once you have your reference genome set up, what you need to do is you need to grow out your F2 population, and you then segregate out the plants that behave like a wild type, and you segregate out the plants that behave like mutants. So you need about 30 individuals for each population. And what you do is you take equimolar amounts of each individual's genomic DNA, and then you pool that into 1 sequencing library, and then you sequence those 2 populations. What you get back is a big data set full of short reads of about 100 base pairs, and then you align those reads to your reference genome. So basically, at the end of the day, you end up with 3 data sets, 1 reference genome and 2 populations which have pooled individuals. And the downstream analysis is fundamentally looking for a location in the genome, which in the wild type pool, for every 4 reads at that location which match the reference genome, 2 of those reads will not match the reference genome. And for the causal mutation and the location of the causal mutation in the mutant pool, none of the reads will match the reference genome. So I'll try to make this a little bit clearer. Basically, what the analysis pipeline is doing is it goes through and it looks at every single polymorphic location between the 2 data sets, and it just makes a ratio of how many reads stack up and match the reference sequence in the wild type, and it makes a proportion of that, and then it subtracts off the amount of reads at that same location that match the reference sequence in the mutant pool. So just as an example, for the allele R that we saw on the previous slide, which is like our ideal situation for what the causal mutation would look like, these calculations are much messier in reality, but this is just kind of a clean example. You would expect to see about 66% of the reads at that location to match the reference genome, and at the mutant location of that same area, you would expect none of the reads to match the reference genome. And the analysis pipeline just zips through the whole data set and makes this calculation for every single location between the 2 data sets. So at the end, what you end up with is basically a linkage map. So the x-axis is just the position in the genome, and here on the left is the output of that calculation from the previous slide. And the program basically just maps all of the locations. So you see a lot of different polymorphic sites here. That's because, as I mentioned earlier, whenever you do the EMS treatment, you end up with a lot of background mutations that are not really linked to your phenotype of interest. And those background mutations end up being fundamentally genetic markers that you can then use. So you can see here on chromosome 3, you have this big peak, and at the apex of that peak is actually your causal mutation. And all of these little sites and all of these alleles that are labeled on the periphery of that peak are linked regions of the genome. So once you have all of that identified,
10 minutes.
So hopefully in the coming months, I will have graphs just like the one on the previous screen for all of my dopamine resistant mutants. And then once you identify the genomic region, or if you have a pretty good idea of what the causal mutation is from these data sets, what you then do is you go through and I'm going to need to do a complementation analysis, which basically entails putting the wild type gene back into the mutant and ensuring that the mutation is actually causing the phenotype that you think it is. And then I hope to use all of this information to try to piece out some part of a working model of how dopamine interacts with plant physiology. So that's kind of concludes what I have for everybody today. But at this point, I'd like to thank everybody in Dr. Jan Smalley's lab and everybody who wrote me recommendation letters to help get this work published. So Dr. Coyne, Dr. Downey, and Dr. Geneve, I'm extremely grateful for your help. And thank you all for listening. I'll take any questions now.
Thank you, Tim. We have some time for questions. You're welcome to write anything in the chat box or take your voice, unmute yourself. We have one question. What is the basis of the sample size of 30 in the F2?
That's a good question. So I think if I recall correctly, if you end up with too small of a sample size, you end up with sort of a lower resolution linkage map. So if the sample size is really small, it makes it a lot harder for the algorithm to actually piece out this map. So basically, you get more randomness in the statistical model. So you need a lot of samples. And it also protects against accidental inclusion of non-wild type phenotypes in each of the pooled populations. It makes it less of a big of a deal if you accidentally include a couple of samples that maybe weren't as well phenotyped as maybe they should have been. I think that this was determined in silico though, if that answers the question at all, hopefully.
There is another question. You mentioned 4 to 2 for the wild type and 0 to 2 for mutant. What does this in terms of segregation?
Oh, so this is something that threw me off too when I was going through this the first time. Really, once you have these pooled populations, since it's pooled, really what the analysis pipeline is looking at is the proportion of haplotypes. So we have these 4 haplotypes here. We have 4 big R's and 2 little r's. So that's what the analysis software is actually reading, because it's reading the proportion of reads. In reality, whenever it's making these comparisons, you have hundreds of reads stacked up at these different locations, and they just roughly end up having a 4 to 2 ratio just because of this fact that whenever you have the wild type pool, you end up, if you just look at the haplotypes, you end up with a 4 to 2 ratio. Hopefully that answered your question.
Okay. Yeah, looks like we are getting to the end of time. If there are any other questions for our team, just feel free to continue in the chat box. And thank you, Tim. You may finish and sharing your screen, and we will move to the next speaker. The last speaker in this session is Gia Tan. Okay, Gia, you should be able to share your screen. We can see everything.
Okay, awesome. Can everyone hear me?
Yes, and we can hear you.
Okay.
Good luck.
Thanks. Good afternoon, everybody. My name is Jia. So today I will be presenting about the results of one of my research, which entails studying grapevine, the regulation of gene expression in grapevine under stress conditions. So a brief outline of my presentation for today. I'll give you a brief introduction followed by my research questions, and I'll go into a little detail about my materials and methods and explain results, conclusions, and finally conclude it with my ideas for future works. So a brief introduction. So as the title suggests, that the plant species I'm working with is grapevine. As many of you know, grapevine is a long-lived perennial plant, and similar to other crops, climate is an important factor in determining the characteristics of this crop. So in the case of grapevine, local climate is usually determines the suitability to grow a particular variety, and it also determines the composition of the fruit that produces, the grapevine produces, which is grapes. And so the change in local climate is usually the source of various biotic and abiotic stresses. And in this case, drought and heat stress in particular are two of the major abiotic stresses that reduces grapevine's yield and the grape quality. And it's predicted that there will be more frequent and widespread drought stress, like drought in general with heat waves, which might lead to, current plant cultivar or variety no longer suitable for local production. So different from like other crops that are like annual crops that are planted around Kentucky, it would be costly for vineyards to grow a new variety because the current variety might not be tolerant or adapted to the drought and heat stress because it takes a long period of time for grapevine to grow and produce grapes. So plants have many ways to adapt and tolerate the change of local climate and the resulting stress. So two common mechanisms that scientists study are usually the physiological changes and the gene expression regulation. In terms of gene expression regulation, people use RNA sequencing technology to study. However, in most RNA sequencing research are focused on model species such as Arabidopsis and some annual crops. The research in RNA sequencing in perennial plants is less studied compared to other things. So for my research, we actually did a comparative analysis, we studied physiological changes and gene expression changes in grapevine under heat, drought, and combined stresses. And combined stress includes heat plus drought stress. So my research questions are how heat, drought, and combined stresses affect plant physiology and how gene expression is affected by heat, drought, and combined stresses. And hopefully in answering this research question, we can identify some differentially expressed candidate genes that might play important roles in the tolerance for heat, drought, and combined stresses tolerance. So our experiment, we used 95 Cabernet Sauvignon plants, and they're divided into 4 groups in 5 replicated blocks. So those 4 groups including control, drought, heat, and combined stresses. And we have taken samples in 6 different sampling times. And I will be showing a graph that resembles more, explains more what those sampling time means. So, and for plant phenotyping or physiological changes, we gather data to measure leaf temperature the changing leaf temperature, stomata conductance, and stem water potential. And we extracted total RNAs and genomic DNAs to study the regulation of gene expression changes. So here's the graph that just to show a clear picture of what those sampling time means. So in sampling time 1 is the plant acclimation period, where the plants first move to the greenhouse with stabilized air temperature, and sampling time 2 to 4 are the stress period. And so for 2 is drought stress only, and sampling time 3 and 4 are drought stress, heat stress, and combined stress together. And as you can see, there is an increase in air temperature during heat stress. And those plants treated for heat stress will move to a different greenhouse. And so time point 5 is the stress removal, and time point 6 is the plant recovery. And we measure air temperature and stomata conductance just to show that, how plant is behaving in those times. So next is just a workflow of the RNA sequencing and the subsequent bioinformatic analysis. So today I'll be presenting some results for some phenotypic results, and a multi-dimensional scaling for gene expression patterns. And I will also be showing some differentially expressed genes and some interesting results based on my gene ontology study. So the first results are the plant physiological changes. As I mentioned in the material and methods, we measure stomata conductance and water potential and leaf temperature. So in the top left graph for stomata conductance, you can see that in general that under heat stress, plants usually have more stomata conductance, which they has higher respiration rate to reduce the intercellular temperature, while drought stress and combined stress reduces the stomata conductance to prevent water loss. So for stem water potential, it's expected that they're all lower, they all have reduced stem water potential compared to control, while leaf temperature combined treatment and heat treatment increases the leaf temperature as compared to drought stress only and control. So next I'll be showing you the RNA sequencing and gene expression regulation part of the data. So we ran a multi-dimensional scaling plot, and this plot just to show you the similarity and differences in gene expression pattern. Based on the pattern, we grouped those into 2 large groups. As you can see, the gene expression at recovery and pre-stress are grouped on the upper corner of this plot, while the stress and stress removal gene expressions are grouped on the bottom. That just to show you there is a difference in gene regulation patterns between non-stress and stress plant samples. And next, I identified some differentially expressed genes based on treatment groups, and the graph shows different numbers of differentially expressed genes in different sampling time. So based on this table, there is a significantly more differentially expressed genes under combined stress than individual stresses. So here is a heat map that shows you. So I think the key point of this page is just that in a combined stress, there are significantly more differentially expressed genes than the individual stresses. And I did a gene ontology analysis afterwards, and I again combined all the gene ontology significant gene ontology terms based on stress. And there is a significantly more GO terms for combined stresses than individual stresses. So the results I'm showing are the combined result. Next, I wanted to focus on one specific sampling time, which is sampling time 5, during stress recovery. At this sampling time, we discovered some very interesting results. So with all the gene ontology terms, we decided to do a network visualization to see how those terms interact and or with each other. So first is the sampling time 5 under drought treatment. As you can see, the network visualization is pretty straightforward or simple, or it seems to be behaving in an additive way. And it is similar for heat stress. However, okay, thank you. However, when you move to the combined stress, you can see the network is more complex, and all those terms seem to be act in a more synergistic manner instead of a more simple additive way. And and when I was looking more deeper into those terms, I found there are some terms that suggested epigenetic changes during stress recovery. So even though we've seen a lot of differentially expressed genes and significant goal terms for all other sampling points, but in stress removal, it's the first time we're seeing genes that are associated with epigenetic changes, so such as histone modification and methylation in general. So to show you a clear picture, we use a visualization tool called Revigo. This is a Revigo graph of the significant gene ontology terms for combined treatment, which is at stress removal. As you can see, some goal terms related to epigenetic changes, such as protein alkylation, protein methylation, protein folding seem to be significant. So we thought that this is very interesting because this is the only time point that shows that. So to conclude, so to answer my research questions, that how gene expression is affected by heat, drought, and combined stresses. Well, for individual stress, the gene regulation seem to act in a more simple or additive way, while for combined stresses, it's more complex or act in a synergistic way. And I think the most interesting thing is that differentially expressed genes associated with epigenetic changes were identified under combined stress as stress removal. This could suggest a potential establishment of epigenetic memory after stress removal. So for my future research, we want to test, can the change in gene expression occur under heat and drought stress be explained by epigenetic mechanisms? So we plan to do this to study through the whole genome bisulfite sequencing to study the DNA methylation patterns under those stress conditions. And hopefully we can find out whether there is a epigenetic memory by doing the same thing for those plants exposing to subsequent stress events. And with that, I would like to thank my advisors and my committee members and the environmental epigenetic and genetic group members. And with their help, I was able to do this, analyze the data set. Thank you, and I'm happy to take questions.
Thank you. Are there any questions for Gia? You're welcome to speak up or write them in a chat box. There is a question. Why study DNA methylation specifically when your geo terms suggested other epigenetic mechanisms might be involved?
Okay, so the reason why we are studying hoping to study DNA methylation is that if we recall from my material, we extracted genomic DNA. So for this data set, we only have the data available to study DNA methylation. So if I would to study other epigenetic mechanisms, I will have to do the experiment again and study, say, like histone modification and stuff. So.
Are there any more questions? Maybe I'll just ask you for clarification. I'm not sure if I understood correctly. Were you saying that when there is a change in gene expression in a stress, when you're taking stress out, the gene expression is not getting back to the previous before the stress.
Are you talking about this?
Not necessarily. I was talking about that that plot that you were on another slide. It was later. It was somewhere at the end.
Oh, at the end.
Mm-hmm. Oh yeah, here. I'm just not sure if I understood that well. If you were saying that the gene expression changes under heat stress, for example, when you remove the stress, does it mean that, did you mean that the gene expression is not the same as before the stress?
I'm not sure, because we were just studying, we were just to identify, differentially expressed genes. I don't know like how many of, well, actually I had a slide in my, like the longer version of this to show you all the overlapping this differentially expressed genes among, like different treatments and different sampling times. I haven't really looked at it, so I can't really tell you whether there were the same, genes were expressed during those times. I don't know if that's clear.
Oh yeah, thank you. Okay, so for the sake of time, we have to finish, but I see there are 2 other questions for you in a chat box, so would be great if you could answer them. And we are at the end of this session. There's going to be 15 minutes break. I don't know, Dr. Yunrin, would you like to add something for the end?